Ever wondered how does the body produce energy for us daily? If yes, let's take a look at this upcoming video. Assalamualaikum and hi everyone. Glycolysis is a process to generate energy in cells where the breakdown of glucose into pyruvates, ATP, NADH and water takes place in cytoplasm of a cell. This process does not require oxygen but occurs in aerobic and anaerobic state. Before we begin, we need to understand the two types of metabolism, anabolism and catabolism. Anabolism is the type that uses energy to construct components of cells like protein and nucleic acids, while catabolism is the type that breaks organic matter and usually harvests energy, for example cellular respiration. So what happens in glycolysis? Now let's take a look at the various reactions involved in the glycolysis. So it all begins with a molecule of glucose that enters the cytosol, and the first reaction that takes place is conversion of glucose to glucose 6-phosphate. The enzyme that catalyzes this phosphorylation reaction is called HEXOKINASE. The enzyme HEXOKINASE transfers a molecule of phosphate from ATP to glucose and converts glucose to glucose 6-phosphate. In this reaction, ATP is converted into a molecule of ADP, hence it utilizes one of molecule of ATP. Next, the glucose 6-phosphate is converted into its isomer, the fructose 6-phosphate, by the enzyme ISOMERASE. This is simple rearrangement reaction. In the next step, the fructose 6-phosphate is converted to fructose 1,6-biphosphate. There is addition of another phosphate group as the first carbon of the fructose ring. This reaction is catalyzed by the enzyme phosphofructokinase, another kinase enzyme which transfers a group of phosphate from ATP to fructose 6-phosphate, and again ATP is converted into ADP. Hence, another ATP is utilized in the reaction. Next, the fructose 1,6-biphosphate is split into two molecules, the glycerol dihydrophosphate and dihydroxyacetone phosphate. These both are 3-carbon compounds, and the enzyme that catalyzes this reaction is called aldolase. These are reactions compose the first phase of glycolysis. The two compounds glycerol dihydrophosphate and dihydroxyacetone phosphate are actually isomers and can be interconverted into each other by the enzyme isomerase. Also keep in mind that every molecule of glucose is split into two molecules. Every reaction of the second phase happens twice and produces twice products. In the next reaction, that glycerol dihydrophosphate is converted to 1,3-biphosphoglycerate. This involves addition of a phosphate group at the first position of the biphosphoglycerate. The enzyme that catalyzes the reaction is the glycerol dihydrophosphate dehydrogenase, and in this reaction, nicotinamide adenine dinucleotide, which is the coenzyme, is reduced to NADH. The 1,3-biphosphoglycerate is converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. This kinase enzyme transfers the phosphate group at the first position of phosphoglycerate to adenosine diphosphate, and in this process, a molecule of ATP is generated. Next, the 3-phosphoglycerate is converted to 2-phosphoglycerate. This is a simple rearrangement reaction that happens with the help of enzyme phosphoglycerate mutase. Next, the 2-phosphoglycerate is converted to phosphonol pyruvate. The enzyme involved is enolase, and the magnesium ions are utilized in this reaction. In the final step of the glycolysis, the phosphonol pyruvate is converted to pyruvate by the enzyme pyruvate kinase. The phosphate group at the phosphonol pyruvate is transferred to ADP and converted to ATP. And that is all for our glycolysis process. Thank you for tuning in and watching. Goodbye!